Okay, continuing on, what if you don't know the genotype for the organism in which you are examining? Well, there's a way to figure out the genotype by looking at the traits of the parent with the unknown genotype and the traits of the offspring. So say you're looking at a purple flower. What's its genotype? Well, can you know for sure? Because purple's the phenotype. How many genotypes can be purple? That's right, homozygous dominant or heterozygous. Both are purple. How do you know if that purple flower is homozygous dominant or heterozygous? Well, we only know one allele for sure, the dominant allele. So we will call it P underscore. So P blank. So we know the dominant allele, and then the second allele is unknown. How do we figure out if it's homozygous or heterozygous? We cross it with a white flower, a homozygous recessive. So P blank times, so our dominant times our recessive, and let's see. So we're doing a test cross. So here's our possible options here. Our P blank could either be homozygous dominant or it could be homozygous recessive. Our second parent, our white parent, is homozygous recessive. So we can have homozygous dominant or heterozygous for our unknown genotype parent, homozygous dominant or heterozygous. And then our known genotype parent is a white flower. So that's easy because you can only be a white flower if you're homozygous recessive. Bam, easy. So let's do the crosses here. Bam. This one, we get all heterozygotes. This one, we get 50% heterozygotes, 50% homozygous recessive. So in the first test cross, where our unknown parent is always purple, the offspring are always purple. In our second test cross, if the unknown parent is heterozygous, half the offspring are purple, half the offspring are white. So what's the genotype? Well, if your test cross produces even one white flower, what is your genotype? Look to the previous. That's correct. Your genotype is heterozygous if you get a single white flower. It's because the only one of these two that could produce a white flower is the one where the unknown genotype was heterozygous. There's the white flowers. No white flowers here. So, easy enough. If after many test crosses you never get a white flower, you can be reasonably confident it's homozygous dominant. However, you can never be absolutely sure because the laws of probability are in play here. And probability means it's random. So there is in fact a random chance that in 20 test crosses you could never get a white flower because, well, 50-50 chance of any particular offspring being purple or white. You could just suddenly hit a streak of purple flowers. So you're never 100% sure if after many test crosses you never get a white flower. However, you are 100% sure if after your test cross you get one white flower. So a pedigree is sort of a family tree of genetic traits. So you can use what you know about test crosses to figure out your family's genes. What you do is you make a family tree showing phenotypes and genotypes, and that family tree is called a pedigree. How do we do it? Well, you identify Mendelian traits in people. For instance, freckles are dominant to no freckles, call it capital F, lowercase f. Widow's peak, where your hair comes to a point at the center of your forehead, that's dominant to no widow's peak, like I have no widow's peak. So capital W, widow's peak, dominant to lowercase w, no widow's peak. And then finally we have free earlobes like mine, where your earlobe can be pulled away from the side of your face. See that? Flick, flick, flick. Pull it away. It's a free earlobe. It's dominant to an attached earlobe, where the earlobe is sort of fused with the skin on the side of your face. So These are some good Mendelian traits. We'll go with earlobes because I don't have freckles or a widow's peak, and none of my siblings do. So let's make a pedigree. My mom has attached earlobes. We know her genotype because you can attached earlobes are recessive. The only way you show the recessive phenotype is if your genotype is homozygous recessive. Easy. My dad has free earlobes. What's his genotype? We don't know for certain. It could be homozygous dominant or heterozygous. So now let's look at the kids, me, my brother, and my sister, in order to see if we can't figure out the genotype. Here we go. Mom, homozygous recessive. Dad, not sure. One dominant allele. I have free earlobes. That doesn't help because there's the dominant allele. My brother has free earlobes. Not so helpful. We still don't know what dad's allele could be. However, my sister has attached earlobes. So we know her genotype is homozygous recessive. Now here's where it works out. Because remember, you get one allele from mom and one allele from dad. In order for my sister to be homozygous recessive, my dad must be able to pass on a recessive allele. So dad, knowing my sister has attached earlobes, is heterozygous. We can go one step further here. We know I got the dominant allele from my dad. So what's the allele I got from my mom? Recessive. So I know me and my brother are heterozygotes. So it's pretty cool. You can work out genotypes by simple things like this. It's really neat. Try it for yourself a few times. It's kind of addictive. It's kind of fun to work this puzzle. So we know this now because my dad produced a recessive trait in my sister. So heterozygous. But let's work it out again just to see it. Here's the Punnett squares. If dad were homozygous dominant is here, and if dad were heterozygous is here. Here's mom with her attached earlobes, homozygous recessive. And here's the possible outcomes. Bam. Here, this one, you can only get free earlobes. Where in this one, you can get free earlobes or attached earlobes. Knowing my sister has attached earlobes automatically means it's this square here, which means my dad is heterozygous. And since I have free earlobes, I automatically know that I am heterozygous as well, since these are the only possible options here for having free earlobes. Neat! Okay, so here's an earlobe pedigree. What are the genotypes of the kids? Sister attached, easy one. Brother, free earlobes. So, I just did this earlier. What allele did dad donate to my brother? Well, the dominant allele. So what's the only allele mom can donate? The recessive allele. So the brothers are both heterozygotes. And here's how you actually write out a pedigree like this. Males are a filled-in shape, in this case a black square, and then I write down the genotype there. So, bam. Kind of neat. You guys should really build a couple of pedigrees. They're really fun to do. All right, now let's talk about the inheritance of genetic disorders, because there are certain genetic disorders that are inherited as dominant or recessive. A lot of genetic disorders are recessive. So if you have a recessive genetic disorder, heterozygotes are considered carriers. Because they have a dominant allele, they do not express the disease. They appear to be perfectly healthy because, well, they're perfectly healthy. That recessive allele is getting ignored because there's a dominant allele. But they can pass the recessive allele off to a child. So if you have two carriers having kids, there's a 25% chance their child could have the disease, the disorder. So, carriers are not affected by the disease. Here we go. Albinism is a recessive disorder. Albinism is a total lack of pigment in the body. You appear very, very pale white. Your eyes are pink. Your hair is essentially white because there's no pigment in your hair. Cystic fibrosis, another recessive disorder. Sickle cell anemia, recessive. That's sort of a special case there because in this case, heterozygotes have normal and deformed red blood cells. And sickle cell anemia is cool because the deformed red blood cells can't be infected by the organism that causes malaria. So you actually have resistance to malaria if you have sickle cell anemia. And what do you know? Sickle cell anemia provides a benefit if you live in a place that has lots of malaria. And when they examined the genes of a lot of African tribesmen, they found that many of them were heterozygotes because the carrier, in fact, gets a benefit. They're protected against malaria. So there's also dominant disorders. Dwarfism, achondroplasic dwarfism, is a dominant disorder. Huntington's disease is a dominant disorder. So there's no carrier for a dominant disorder because there's no way to have two different alleles and not express a dominant disorder because, well, it's dominant. If you have a dominant and a recessive, you express the dominant. But what's interesting about this is if you have two dwarf parents and they both are heterozygous, there's a 25% chance their child could be of normal height. So kind of neat little side effect of dominant disorders. Huntington's disease is a lethal dominant disorder, but it tends not to show itself until late in life, meaning you're in your 30s, and it's possible to have kids when you have Huntington's disease and not know it. And when you have kids and you have Huntington's disease, unless you're heterozygous, you're passing on a death sentence. If you're heterozygous and you have kids with someone who doesn't have Huntington's disease, there's only a 25% chance your kids won't die from Huntington's disease. It's kind of one of those scary ones because you can be tested for it. But, I mean, what are you going to do if you have it? Will you decide not to have kids if you're heterozygous? Because there's only a 25% chance your kids would live a full adult life? Will you have kids anyway, even though their lifespan will be shortened? Up to the particular person. So when you test for disorders, you can do for a fetus an amniocentesis, in which case you take a sample of amniotic fluid from the uterus of a mother with her baby growing. And there are fetal cells in the amniotic fluid. When you grab a hold of those, you can sort of wait for them to undergo mitosis and then freeze them during mitosis and look at the chromosomes. So you take a sample of amniotic fluid and you test those fetal cells for aneuploidy, meaning monosomy or trisomy. You can look at the number of chromosomes and determine if you've got a problem with the number of chromosomes. Then you can take this one step further and go full-on genetic sequencing, in which you find out the specific sequence of DNA in a person. All the A's, G's, C's, and T's, man. So there we go. We know the genes for some genetic disorders. So you can get your DNA sequenced and find out if you have a gene for a genetic disorder. And we'll come back talking about some wrench in the gears of Mendelian inheritance. Because, in fact, not everything is inherited the way Mendel discovered.